Welcome, sir. This is Sandipan here from the Botany Department. Hello, sir. Can you hear me? Your mic is off, I
Okay,
guess.
yeah, please, please. Yeah, it's
Yeah, yeah,
audible. Thank you, thank you.
yeah. Welcome, sir. We will wait for 5 minutes to other to join. Then we
Yeah,
can start, sir.
okay, okay, no issue.
Students are joining. Okay. Uh, Good afternoon, everyone, and welcome to this DBT Star College scheme. an online webinar on molecular techniques for the UG students. So, with the permission of our principal sir, honorable principal sir, I am starting the session. And on the very beginning, I would like to request our honorable principal sir, Dr. Devnath Palit, to say a few words
Okay.
regarding the program. So, please.
Good afternoon to you all. Thank you, Sandipon. Thank you very much. So, I welcome you all to this invited lecture organized by the Department of Botany of Durgapur Government College. And first of all, I would like to express my deep sense of gratitude. And really, I'm grateful to Dr. Zahed Hossain, Professor, Department of Botany, Kalyani University, for accepting our invitation and for his kind consent to deliver a popular lecture on the topic as mentioned by Sandipon sir. So this invited lecture, I think, on the modern techniques of molecular biology and plant biotechnology will introduce the fundamentals of the topic to undergraduate students to achieve the objectives of practical training and knowledge of handling the experiments under the DBT Star College scheme. And students, you are very lucky enough. You have with you is such an eminent resource person in the field of particular biotechnology, plant biology, etc. I know him very well. So this will induce the sense of experiential learning and motivating them to pursue life sciences as career. Myself being a student of botany, so I am eagerly waiting to hear from Professor Ocean. Without much delay, I wish a grand success of this seminar. And my earnest request to the students: please interact after completion of this seminar, and this will be the most enriching and interactive session. Thank you so much.
Thank you so much, sir, for your kind words. Now, I would like to request our TCS sir, who is at the same time one of my colleague in the Department of Botany, Dr. Shubhajit Oja, to please say a few words regarding the program.
Uh, Thank you, Sandipan sir, respected principal sir, Durgapur Government College, and respected coordinator madam, DBT. Today, I think this is the best opportunity for the students because our most favorite Jayed sir is here in this online platform. I have met him in last on his marriage ceremony. So far I know he's very humble person and very much highly efficient in this field. So students, especially the sixth semester, you must listen to sir and interact with sir so that you have any questions, please interact. And on behalf of the Durgapur Government College Teachers Council, I heartily welcome Jayed sir in this online platform and all the participants present over here. And this is mainly for the UG students. So students, please you just listen to sir and interact sir if you have any questions. So I wish this seminar would be a great success. Thank you so much, Sandeep sir. Okay.
Okay, shall I start or?
Yeah, but just a little formality, sir.
Okay.
Actually, I requested our head of the department, Dr. Ashok Bhattacharya, to introduce you for the student. Just a little
Okay.
formality, sir.
Thank you very much. Actually, Professor Zahed Hossain, he is self enough to introduce himself. But I know him for last 22 years. And as I know him personally for last 22 years, just I'm very happy to say something about him as I have got him in this dais now in the online platform. So Professor Zahed Hossain, when I was introduced at first time, then he was Zahed Hossain only, not the doctorate. He was as the JRF, Junior Research Fellow. So Dr. Zahed Hossain, he has a several teaching and research experience, more than 18 years, so far I remember, more than 18 years teaching and research experience he has gathered in the field of the plant genomics, plant proteomics, microRNAomics, nanotoxicology, and he has gathered many awards at the national and the international level. Just I'm willing to mention a few of them. He has a lot of publications at the, if you see the Google Scholar, the Orchid, everything, then you see about the details about that. But I'm not mentioned about the detailing. A very high impact factor publications always he do, and he is carrying out the research as well as different research project he's doing on, taking the fund from the central DST, central DBT, CSIR, and in the University of Poland now he is the one of the eminent researcher. So I know, and of course all these informations are available in the website and in the internet. But in the field of the plant genomics, plant proteomics, microRNA omics, nanotoxicology, he is doing a lot of work. So Professor Zahed Hossain, he did his undergraduate BSc from the University of Poland in 1998, securing first class first position, gold medalist. In 2000, he did his MSc from the same university, University of Poland, securing first class first position, gold medalist. Then he qualified CSIR NET in 2000. Again, he qualified CSIR NET in 2002. And in 2002, he secured the position in top 20% of the CSIR NET. And he was the subsequently recipient of the CSIR Junior Research Fellowship and CSIR Senior Research Fellowship. And I know that he did his PhD from the CSIR NBRI Lucknow, when I spent a lot of days and the night with him at the same hostel, CSIR hostel at NBRI Lucknow. And I don't forget, always I remember that days that I have spent with Zahed, everything, the lunch, dinner, everything with Zahed. Anyway, so then he got the job in the College Service Commission, and WBCSC, West Bengal College Service Commission, for consecutive 2 times he was the topper in the West Bengal College Service Commission. Then after getting job in the college, probably in the Noihati or something, just a few years back, he joined in the West Bengal State University as the faculty member. Then he moved from the West Bengal State University to the University of Kalyani. And interim period at the University of Kalyani and the State University, he has visited a number of the country and he got a number of the award. Visiting researchers, he did his postdoctoral research as the visiting researchers in Ecophysiologia, Biotechnology, University of the Jaume, so far my knowledge is concerned, Spain. Probably it was in 2007. Then in 2008, he was the recipient of the DST Fast Track Young Scientist Fellowship. Then the visiting researcher, Ecophysiologia, Biotechnology, the same country, Spain, 2009. And he again he did the work in the as the DST Boyscas Fellow, probably in 2010. Yes, 2010, 2011, DST Boyscas Fellow. Better opportunities for the young scientists in choosing areas of the science and technology program in the Department of the Science and Technology. And he did the postdoctoral research as the DST Boyscas Fellow in Japan. And not only that, he got the JSPS fellowship. It is one of the prestigious fellowship in the world, you know, and he accumulated this award also in his bag. JSPS, Japan Society for the Promotion of the Science, Invitation Fellow, 2014, short term, the national, and he did the work in the National Institute of the Crop Science, Japan, and in latest time he accumulated the prestigious award from our Department of the Higher Education, Government of West Bengal, recipient of the Shiksha Ratna Award in 2021. So these are the something. Not only these, many other awards and the achievements Dr. Zahed Hossain he has gathered and he has done. So I think that the students, you will also get the information and you will be inspired. A lot of inspiration you will get from the Zahed sir, because Zahed sir was also a same botany students like you once upon a time, and I have seen him as the botany research scholar, and that's why I'm very much thankful on behalf of the Department of the Botany and on the full encouragement receiving from our respected principal sir and the DBT authorities, DBT central DBT authorities that we are getting opportunities for this, and our coordinator madam DBT Star College scheme, she is also trying to extend her hand. As such, we can accomplish such type of the events and program, so that the students can be benefited a lot from the academic point of the view. And that's all. I'm very much thankful, Zahed, because we are getting you in this platform to say something and to share your knowledge, your vast knowledge. But our participants are very minor, and just try to give something from your behalf. Thank you so much.
Thank you. Thank you.
Thank you
So,
so much, sir. Guy said the stage is yours. You can start your
okay.
presentation.
So first of all, a very good afternoon to all of you. So it is indeed a pleasure for me to deliver this lecture on this online platform after a long time, because after lockdown, this may be the first lecture that I'm going to deliver on this platform. And I express my sincere thanks to Dr. Devnath Patil, the principal, Durgapur Government College, for inviting me, sir. Thank you. And I'm also thankful to the organizer, the coordinator, madam, Dr. Shondipon Roy, Dr. Shubhajit Oja, and head Department of Botany, Dr. Ashok Bhattacharya, and all my respected colleagues of Department of Botany, Durgapur Government College, and my fellow students. And I'm as Ashok Da. Actually, I used to say as Ashok Da. He is basically a senior Dada, and also philosopher and guide, because I spent a very good time. We had a very good time at NBRI. We spent there, and that time I was doing my PhD research work, and Ashok Da was doing his fast track scientist fellowship. With this, he was doing the fellowship, this research. And what Ashok told, that I had spent maybe 2 or 3 years with him there, and whenever I was in a trouble, I used to go to him for his help and his guidance. So he was always generous to give me that kind of suggestion. So thank you, Ashok, for inviting me. Without wasting time, I would like to start my lecture. So for today's lecture, I have selected, since the audience is basically UG students, so from their syllabus, I have just selected a topic that is all about plant tissue culture. So just so is it visible, or am I audible? Is it okay? So,
Yeah, it is visible, sir, and
and
you are audible also, and it's moving also.
okay,
It's
then
moving.
it's fine. Okay. So thank you. So students, I have selected a simple topic, that is a plant tissue culture technique. Although it's very vast topic, today it's not possible to cover all the areas or all the aspects, but I would like to give you a bird's eye view on plant tissue culture techniques, what are their applications, and what are the different aspects of plant tissue culture. So before that, we must know what is plant tissue culture. So you know that culture of plant cells, maybe tissue, maybe organ, or even seeds or protoplast, so on the nutrient media. So you have to culture these either cells or tissue or organ or seeds or protoplast or nutrient media, but under aseptic condition, because you can't do a plant tissue culture at your home or at your class lab or like that. So you need a specific area which should be a aseptic area, and for that you need certain instruments, or you can say equipments. So the overall definition would be the culture of plant cells, tissue, organ, seed, or protoplast on nutrient media under aseptic condition, so that you can, which lead to the cell multiplication. That means it will lead to the formation of callus or the regeneration of the organs of the whole plant. So that is the complete definition of plant tissue culture. So you know that a tissue is composed of so many so many cells, and similarly the organ, it is made up of several tissues. So all these, so any part of plant can be used as a explant. So here I use the term explant. Explant is nothing but the starting material. So that any part of a plant that can be used to initiate the culture is known as a explant. So here it may be a plant cell, it may be tissue, it may be organ, it may be even seeds or protoplast. So you must be wondering why I say a seed, because seeds normally germinate, but we can, I mean, establish culture using seeds as an explant for those seeds which are basically non-endospermous, particularly for the orchids, orchid seeds, you know, these seeds are non-endospermous. So it's very difficult to germinate. So it in nature, it basically takes the help of some fungus so that it gives the nutrient. So, but in nature, if you just throw the seeds, it will not germinate because there is no endosperm. So for that, embryo culture is one of the technique to multiply orchids. So that's why I mentioned the term that is seed. So seeds can also be used as a explant or even the protoplast. So this is a basic definition of a plant tissue culture, and you should remember the name of the father of plant tissue culture, that is the Gottlieb Haberland, because he in 1902 he first attempted the culture of mesophyll cells and root hairs of Lemium. So that's why he is coined as a father of modern plant tissue culture. So, so the plant tissue culture, it basically relies on these three fundamental abilities of plant cell. That's very most important. The first one is a totipotency, second one is a dedifferentiation, and the third one is a competency. So, totipotency is nothing but the inherent capacity of plant cell or tissue to develop into an entire plant. So, only the plant cell, they have these potential to, if it is stimulated in a certain way or under certain condition, that can be developed into a entire plant. So this is the inherent capacity of plant cell or tissue to develop into an entire plant cell, entire plant, that is known as a totipotency. And the second one is a dedifferentiation. That means this is again the capacity of a plant cell where the mature cells is return or revert back into meristematic condition and that develop into a new growing point. So dedifferentiation is also basically a fundamental abilities of plant cell. And third one is a competency that basically describe the endogenous potential of a given cell or tissue to develop in a particular way. So these are the three fundamental abilities of plant cell, or you can say these are the three pillars behind the success of plant tissue culture. One should be totipotency, second would be dedifferentiation, and the third is competency. So what are the advantages of plant tissue culture? So the first one is, all the regenerated plants are genetically homogeneous or genetically identical with the mother plant or the explant, because this technique, once you are culturing a cell or tissue or whatever the explant, all the derived plants would be genetically identical with the mother plant or the plant or you can say the explant. So that is the most advantage of the using the plant tissue culture. Second one is the plants are obviously virus free because you are culturing under aseptic condition. So it is expected that the tissue raised plants are virus free, and it is very easy to maintain large number of germplasm within a very small space. I mean, I will show you some photograph and I will talk on that. So the next point is it's very easy to produce hybrids for incompatible species, because some species are cross incompatible. So for that, so normal breeding method will not be applicable. So for that, it's very authentic, or you can say that the most appropriate method of raising hybrid for incompatible species. Or even you can raise haploid plant. Haploid plants you can raise by using explant, which is haploid in nature. So you can even culture anther as a explant, or the pollen as a explant, or the ovule as a explant. So as the explant is haploid, so all the regenerative plants would be haploid. So this is one advantage of the plant tissue culture. And the most important is a round year production. That means you can produce or you can propagate plants throughout the year. It is season independent. So just like if you see, if you in case of a if you try to grow rice under field condition, you have to wait for the appropriate season. But here you can multiply plants throughout the year. So that is season independent, and it is also a effective way of propagating these plants, which are normally it is difficult to propagate. So for that, plant tissue culture can be applied. And using this, you can raise new variants. So there is only two options is there. One is a somaclonal variation, that is the variation that arises in the somatic cell, and also you can induce mutation. So that is called in vitro mutagenesis. And nowadays it is being used as a research tool to raise the transgenic crop. So these are some of the advantages. So you should remember at least 2 to 3 points. The first should be the genetically identical with the explant or the mother plant. It should be virus free, and you can maintain large number of germplasm within a very small space, within a or you can say in a very small you can say the infrastructure. And but there are certain disadvantages, because it requires expensive facilities, because you need a culture room, you need AC, you need culture rack, you need laminar hood. So it is not a very cost effective one until and unless you are doing a large number of culture. Okay, so it requires expensive facilities. Also, a layman can't do this. So particularly skills, or you can say skilled technicians are required to carry on this plant tissue culture. And there is another problem is, although we are saying that the raised plant or tissue raised plants are genetically homogeneous and genetically identical with the explant, but there is a chance for somaclonal variation that you have to identify. But that needs certain molecular tool. So that's one point. That is the error in maintenance of identity. That means in case of micropropagation, it is expected that all the plants that are raised through this technique are genetically homogeneous, but there is high chance due to the somaclonal variation some changes at genetic level may happen. So identification of that somaclonal variation is very much required, and which requires a sophisticated facilities. So that is one disadvantage. And also on introduction of unknown pathogens. Okay, so that is another problem, because we know that these plants are prone to these pathogens. So we used to test those samples for the pathogen, whether they are pathogen free or not. But sometimes what happens, introduction of certain unknown pathogens may create problems. So these are certain disadvantages. So as I mentioned in my first the slide, the explant may be a cell, maybe tissue, maybe organ, or maybe protoplast, maybe seed, like that. No, so here you on the left hand side you can find this is basically a segment of leaf. That means uh, we used here as leaf segment as explant. So that is basically you can say a segment of organ that is being used as an explant. So from there you can find the two shoot buds they have raised from these leaf explant. That means here the shoot buds can be raised from the explant directly. So based on these, you can differentiate plant regeneration, either it is under organogenesis or it may be embryogenesis. So development of organ, it may be a shoot, it may be a root from the cultured explant, is known as organogenesis. And formation of embryo means somatic embryo, it is known as the embryogenesis. The organogenesis and the embryogenesis, these are the two different types of plant regeneration method or modes. But organogenesis again may be a direct or indirect. When we say a direct, that means when the organ develops directly from the explant without going through the callus phase. But here you can find callus is first formed from this explant, and from this callus the shoot bud raised. So this would be called direct, this would be called indirect. The photograph on the left hand side, here you can find the shoot buds directly regenerated from the explant. That means it should be direct organogenesis. But on the this panel, the below one, here you can find this is a callus. So this callus means undifferentiated mass of cell. So from the callus you can find shoot buds they regenerated. So here it should be called indirect mode of organogenesis, because here organ develops not only directly from the explant, but it is from the callus. So if the organogenesis is without going through the this callus phase, it would be called direct organogenesis. And if it goes through the through the callus phase, then it would be called indirect organogenesis. Similarly, if somatic embryo directly develops from the explant, it would be called direct embryogenesis, or if it forms from the callus, then it would be called indirect embryogenesis. So these are the two different modes of plant regeneration. One is the organogenesis, another one is the embryogenesis. So this is basically the general scheme of establishment of a culture or aseptic culture. So it first starts with a collection of plant material. So the plant material, if it is of big size, then first you can cut the plant material, then you do the surface sterilization, and then we will do the rest of the things. But if the material is the plant material itself is a small one, so there is no need to cut the explant. So first we do the surface sterilization, and then we make some minor cutting, so that is called cut to cut the explant. Then again we do the surface sterilization, and we culture them on this different type of media is there, and the media should be supplemented with different hormones. So it is based on your objective. If you want to induce callus, the hormonal composition would be different. If you induce direct organogenesis, then the composition would be different. So it all relies on the combination of different plant growth regulators. So here the step, if it follows a callus phase, that means the cells, the already differentiated cells, it will follow the de-differentiation pathway, and it becomes meristematic. So it will start growing and multiplying, so it will form a callus. So this callus is nothing but a undifferentiated mass of cell. So from this callus the embryo may develop, so that is called the embryogenesis. Then from the embryo you will find you will be able to grow the whole plant. Or from the callus, organ may develop. Organ means first shoot develop, and then you have to cut the shoot and you have to transfer it in a rooting media. So finally you will get a plantlet, that means shoot with a root. Then you have to go through the hardening process. So this is one step, an essential step for plant tissue culture, because these small plantlets, these are basically newborn babies. Okay, so you have to take care of these babies. So for that, if you directly take out these plants from the culture tubes or the culture vials and directly shift it to field condition or in the net house, so they will not survive. So that's why there is a step called hardening here. Basically that's basically acclimatization process, because the newly developed or newborn these plantlets, they get acclimatized with the surrounding environment, because for this reason the plantlets are being maintained under the same control environment for some days, maybe for 10 to 15 days. Then you can shift it to the net house or glass house, so that the success rate would be higher. So this is in general a scheme of inducing a culture. And on the left side I have mentioned what is explant. In the first slide I mentioned that explant is any part of a plant. That means it may be a cell, it may be tissue, it may be organ, it may any part that can be used to establish an aseptic culture is known as explant. So these are some photographs of different stages of plant tissue culture. So on the first panel, left hand panel, you can find that the two workers, they are, or you can say the skilled technicians, they are doing this culture. So here this particular device or instrument is known as, it's a basically a laminar hood. So I will explain this in later. So this particular cabinet, it provides that aseptic condition. As I mentioned that you can't do your plant tissue culture at your home until and unless you have this system, because this platform will give you the aseptic condition. And these are the different steps of plant tissue culture. You can find this after doing the culture, after inoculation, you have to keep these plants under the in this rack, which are fitted with fluorescent tube light, and they are controlled by some timer. So that will basically mimic the photo period. Usually 16-hour day and 8 hours night is usually it is maintained using the timer. And these are the different culture vessels. This is called jam bottle, or you can say the big jam bottle. This is culture tube, not the test tube, because here the width is much wider as compared to the normal I mean the chemistry culture tube. So, and these are you can find here there are two different types of media you can find. One, the first three are very clear, and the next two is little bit you can say the foggy. So first three we didn't apply any gelling agent. Okay, so that's why it is liquid. So the media may be liquid, media may be solid. It depends on your objective, what you are going to culture. So if you need a liquid media, so there is no need to adding the any gelling agent. But if you want a solidified media, so you have to add agar. So in the media, while preparing the media, and based on the solidification of the position of the this culture tube, the media, it may be slant or it may be stub. Here you can find this is a one picture showing the slant position. That means here you will get large area for the inoculation. That means here you can accommodate at least 3 to 4 explant if the explant size is small. Whereas here you can find it's a stab. So here usually the nodal explant, that means node with the axillary buds, is basically used to inoculate for in these type of stab culture. And this is a photograph showing the hardening step. So as I mentioned that it depends on your objective, what you are going to do, because once you inoculate the egg plant, if you wish to induce callus, then the hormonal composition would be different. If you wish to raise directly this shoot bud from the egg plant or from the callus, then your composition, hormonal composition would be different, because in most of the media the basic composition remains same. Only difference is the this called the phytohormones or the plant growth regulators. So it is mostly this is the balance between the auxin and cytokinin. It basically regulates or determine the fate of your culture. So mostly what happen when there is a high auxin cytokinin ratio, it usually induces rooting, whereas when the cytokinin auxin ratio is high, or you can say the auxin cytokinin ratio is low, then you can find there is a shoot bud formation is generally accepted. Whereas it will also be applicable for the plant Y. So what even our case, what we have found that without adding any auxin, routing is there. So it all depends on the plant type. So every time you have to standardize the protocol. You have to every time you have to standardize the you can say the balance between the auxin, cytokinin, or the gibberellin if it is required, or the abscisic acid. So different hormonal balance is very much required to get desirable, you can say the expected result. So this is the same, you can say scheme. Here it is in case of scrophularia. You can find that once you increase the BAP, that means cytokinin, there is a enhanced, I mean, level of formation of the shoot buds. Whereas if you increase the auxin, here you find the IA or NA, so there would be a higher rate of root formation, unlike this. But as I mentioned, this is not very much, you can say that hard and fast rule. If it is applicable to scrophularia, it does not mean it will applicable for rice or wheat or like that. So, so as I mentioned that in the plant regeneration system, it may follow organogenesis, or it may follow a somatic embryo. So these are the some of the pictures of somatic embryo of different plant species. So this is again some pictures of showing shoot bud morphogenesis, or shoot buds raising from the explant directly, and this is the somatic embryo. So all the photographs are taken under stereo microscopes. So this photograph, you can find that there is a different stages starting from the inoculation, from the shoot bud regeneration. As I mentioned, you find this is called the shoot organogenesis from leaf explant. Since it is direct without going through the I mean this callus phase, it is more appropriate to say that's a direct shoot organogenesis. So once you get But one thing I must mention, that here we just took out this explant, we sacrificed this explant, because once you took out from the vessel, you can't replace it or place it again on that, because it means it becomes contaminated. So for photograph, we basically took this from the culture vessel. So once you get these type of shoot bird, you may proceed for the multiplication, because for multiplication media you will get large number of shoot birds. Then you can shift it to the rooting media. Here you can find the clear roots, they have come up from the shoot birds. So once you get a shoot bird with a root, that means young plantlets, then you can transfer, but through the hardening stage. Here you can find this is the hardening stage, and this you can we shift it to the pot. So here this is basically a chrysanthemum plant, and these two pictures are from the scanning electron microscope of initial stages of the shoot bud formation. Here these are called, these are basically hairs you can find under scanning electron microscope. And as I mentioned, that any part of a plant can be used as a explant. So here you can find, I don't know whether you can see or not, this is basically the ray floret. You know that in case of chrysanthemum, there are ray floret and disfloret. So we used ray floret as a explant, and you can find these young shoot buds, they are coming out from the ray floret. So again, this is a direct shoot organogenesis, because it is not going through the callus phase. So any part of a plant can be used to induce a aseptic culture or to establish a culture, because the plant cell have those three properties. One is the most important is the totipotency. Because of the totipotency, the plants, if it is properly triggered or properly stimulated by those you can say the plant growth regulators, so that can be the cells can regenerate into a new organ or new plant, huh? Or the de-differentiation can be possible, because the cells, because you see that this a ray floret is there, so cells are already in a differentiated state. So once it is a mature state, it should be revert back to a meristematic condition. So that is induced by adding those plant growth regulators and other stimuli. So here you can find that these are basically the callus. Callus is nothing but the unorganized mass of cell. So here you can find these green zone. These are basically the live callus. This is basically the dead callus. So from the callus, we basically induce this shoot bud regeneration. So it should be called indirect. That means because it is going through the callus phase. So this is called indirect shoot organogenesis. And as I mentioned, for even seeds can be used as a explant, so that the classic example is orchid, because orchid seeds are non-endospermous. That means there is no nutritive tissue, so that it will give the nutrition to the growing embryo. So for that, the embryo can be rescued, and those embryos can be cultured under aseptic condition. So that's why seed culture. But if you try to establish a rice culture using a seed, it is of no meaning, because rice seed is usually, it's normally it grows, huh? If you soak rice seeds and if you grow it in a natural condition, the next day or maybe day after tomorrow, the plant will regenerate. So there is no question of doing a seed culture for normal plant. So it is only required for the plants where there is a problem with the germination. So orchid is basically a classic example. So I mentioned that the somatic embryogenesis is one of the mode of plant regeneration. One is a this shoot bud organogenesis. Another one is a somatic embryogenesis. So embryogenesis means is basically again the process of developing somatic embryo. Somatic embryo is nothing but the embryo that is derived from a somatic cell. What you know that when a male gamete and a female gamete unites, it forms a zygote. Then zygote will turn into an embryo. So that is called a zygotic embryo. But here, only from the somatic cell embryo develops. So that embryo is known as a somatic embryo. That means embryo-like structure, it's very similar with the zygotic embryo. These are derived from the somatic cell. So that potentiality or that process of regeneration of somatic embryo is known as somatic embryogenesis. So This embryogenic potential, that means that has to be triggered by using definite combination of plant growth regulators. Until and unless you trigger the somatic cells, it will not turn into somatic embryo. And this is one term that is called embroid. These are basically very normal embryo-like structure that's very similar with the zygotic embryo. We call it is embroids. So this is the development pathway of the somatic embryo. So somatic embryo basically it originates from a single cell, single superficial cell. So that cell is basically start dividing. So why it start dividing? Because it becomes meristematic. Why it is meristematic? Because it undergoes the de-differentiation process. Because all the cells, suppose you are doing a culture of a very small so leaves are already, the cells are already in a mature state. So that mature cells should be revert back into the meristematic one or meristematic condition. So that is known as dedifferentiation. So that dedifferentiation should be triggered by definite stimuli, particularly with the actual, or you can say the very delicate balance between these different type of plant growth regulators. So once it becomes meristematic, it will start dividing. So it will form a mass of cell or group of cell, and that group of cells, it becomes isolated from the rest of the cell through the formation of this cutin. So this cutinization of the outer layer, it basically make them separated from the rest of the cell. Now it again start growing and it will form different development stages, one called globular, then heart shape, then torpedo, and the cotyledon stage. And this somatic embryo is basically the development. If you see the development of the somatic embryo, it is very similar with the zygotic embryo. Apart from this callus, this, or you can say the globular stage. So beyond the globular stage, this heart shape, cotyledon stage, torpedo, all you can find in case of zygotic embryo development. So here you can find this somatic embryo. It is basically a bipolar structure. So on the upside there is a plumule, the downside is a radicle. So you can see the somatic embryos are bipolar. They have a radicle, they have a plumule. Okay. So to induce the somatic embryo, usually very high dose of auxin, particularly 2,4-D, is basically applied to the media, which basically triggers this de-differentiation process. Okay, so but there are certain differences between the somatic embryo and the shoot bud, because shoot bud, you find here the shoot bud is basically a monopolar structure. So that's why I have just incorporated one table just to differentiate between shoot bud and somatic embryo. So in the regeneration protocol, I have shown that the regeneration, it may be through organogenesis or it may be through embryogenesis. So if it is organ, then shoot bud formation is the organogenesis process. But somatic embryo development comes under the somatic embryogenesis. So shoot bud is origin is multiple cell, whereas somatic embryo is single cell. So both are superficial. That means the shoot bud regenerates from multiple cells, multiple superficial cells from the explant. But somatic embryo, it develops from a single superficial cells from the explant. Shoot buds, as I mentioned, it's unipolar. It has only shoot pole, or it's only the upper part. Okay, no radicle is there. But here it is bipolar. Both shoot and root poles are there. But in case of shoot bud, there is a vascular connection is present with the explant, or you can say callus. So that's why. But in case of somatic embryo, there is no such vascular connection is present. That's why, since there is vascular connection is present in case of shoot bud, so it is not very easy to separate until and unless you cut off that shoot bud from the explant. So you have to physically use a scissor to cut a shoot bud, and you have to separate it from the explant or the callus. But in case of somatic embryo, since there is no vascular connection, you can easily separate from the explant or the callus. So these are some basic difference between shoot bud and somatic embryo. This photograph I have already shown. So this is just I'm showing the molecular basis of somatic embryogenesis. I'm not going into details. Only some genes are basically responsible. One is known as somatic embryogenesis receptor kinase, and another signaling pathway, that is the bacillus-steroid signaling pathway, is involved, which basically induce the somatic embryo development. But apart from these ACRK-BR signaling pathway, some group, they believe that DNA methylation followed by chromatin remodeling is basically responsible for this cell genomic reprogramming, which leads to the formation of embryogenic cell. Embryogenic cell means somatic cell. Once you treat with a high dose of auxin, there should be a genetic reprogramming so that the somatic cell becomes embryogenic. So then the somatic embryo can be produced. And also there are some school of thought is there that leafy cotyledon 2, which is basically a transcription factor, that basically induces auxin synthesis, and that leads to the formation of somatic embryo. So these are the different molecular pathways of the induction of somatic embryo. So either you have to propagate through inducing somatic embryo or through inducing the shoot bud. So whatever the mode, there is another application. If you encapsulate these somatic embryos or the shoot bud by some chemical, so that seed would be called synthetic seed. So synthetic seed is nothing but the artificially encapsulated somatic embryos, or it may be shoot bud, even some cell aggregates on any other tissue that can be used for showing as seeds, and that possesses the ability to convert into a plant under suitable condition. That means these are called synthetic seeds because here within this jelly, here it is made up of calcium alginate. So within this capsule there is either somatic embryo may be present or it may be shoot bud. So that somatic embryo will germinate and that will grow into a new plant. So this is a possibility of, you can say, the application of plant tissue culture, where these shoot buds or the somatic embryos can be encapsulated using different chemicals. And so there are basically two different types of synthetic seeds. One is called desiccated synthetic seed, because here the encapsulated material is called polyoxyethylene. So that is basically used to encapsulate that either the somatic embryo or the shoot bud. But here there is another encapsulating material is there, that is called the calcium alginate. So here the seeds remain hydrated, so there is no chance of desiccation. So here those embryos, or you can say those somatic embryos or the shoot buds which are basically you can say the tolerant against the desiccation, here basically desiccated synthetic seeds are being formed. But for those which are very sensitive to the heat or the dehydration, for that hydrated synthetic seed is used, and for that so calcium alginate chemical is used to encapsulate those material. So but what are the advantages of synthetic seed? So that the one advantage should be large scale production. You can produce large number of synthetic seed using either somatic embryo or these shoot buds, because once you induce the shoot buds, you will get large number of shoot buds. Similarly, you will get large number of somatic embryos. So a large number of synthetic seed can be produced within a very short time. And also it maintains genetic uniformity, because all the plants are genetically homogeneous. All the shoot buds or all the this called the somatic embryos are genetically homogeneous with the mother plant or the X plant. That's why it maintains genetic uniformity. And low production cost. You need only one or two chemicals, which is not very costly. And it facilitates rapid multiplication of plants. Definitely, within a very short time, you can produce large number of synthetic seeds. So all are these some of the examples of advantages, but there are certain disadvantages also. That somatic embryos usually have a very low survival rate. So it is sometimes what happens, the synthetic seeds they lose their viability. Okay, and sometimes what happens, inefficient maturation of somatic embryo also lead to poor germination. That means poor growth and development sometimes can be seen in case of using the synthetic seed instead of going through the normal plant tissue culture technique, so that you can raise the whole plant and you can do the hardening and you can shift it to the you can say the field condition or the shed net. So there the success rate is more. And also sometimes what happens, the concentration of the coating material is also a limiting factor, because it basically inhibits the germination of that somatic embryo or the shoot bud. So there is another term very similar to the plant tissue culture, that is called the micropropagation. But there is very thin silver line that basically differentiate plant tissue culture and the micropropagation. So micropropagation should be large-scale production of 2,2 type plants. 2,2 type means it is just a clone, just a Xerox copy. That means 2,2 type of plant. That means it is just a clone of the X plant or the mother plant from which these plants are derived. So micropropagation should be large-scale production. Not if you wish to raise 50 plant or 100 plant, you can't say it's a micropropagation because the number is very, very less. But once your objective is to raise maybe 1 lakh or 2 lakh plants per month, then it would be called micropropagation. So first, large scale production, obviously 2,2 type because it should be identical with the mother plant, it should be genetically uniform, and it should be disease free. So these are some basic points which basically define micropropagation. Although it basically comes under the umbrella of plant tissue culture. Okay. So there is another time we used to use in our, the literature, that is the clone. Clone is nothing but a genetically identical group of individuals. Or you can say these are the genetically uniform, genetically identical with the mother plant is known as a clone. So since it's an asexual mode of reproduction, so that basically maintains this genetic fidelity. An explant I have mentioned that the starting material. So micropropagation, what are the advantage? Very high multiplication rate, because if you do a conventional propagation technique, maybe cutting vegetative propagules, so you may raise maybe 10,000 plant per year. But if you follow micropropagation, you may raise 10 lakhs plant per year from a single explant. So micropropagation, it has a great advantage, particularly for the high multiplication rate. And also very small size explant is required. You can establish a culture from a very small size explant. So that is again an advantage for the micropropagation. And also to maintain these micropropagated plants, maybe you can maintain in a jam bottle, maybe you can maintain in a conical flask. So it requires a very small space. And also packing and transport is also easy due to their small size, because if you transport maybe a jam bottle fill up with this tissue-based plant, so there you can at least transport maybe 30 to 40 plants. So that is one advantage of using the plant tissue culture, particularly micropropagation. And very important, we know that in plant system there are dioecious species are there, particularly for example papaya. So you find papaya, there is a male plant, female plant, and like that. So in this process, using this micropropagation, you can selectively, culture your desired plant, because, or you can say the plants of desired sex can be selectively multiplied. So in case of papaya, it is expected that you will multiply only the female plant to get the papaya. So there is possibility to selectively multiply a desired sex of plant. Okay. And obviously the raised plants are healthy, pathogen free, because it's raised through the this aseptic culture, and easy to export. No quarantine issue is there because it's virus free, and independent of season. I mentioned that throughout the season, throughout the year, you can produce or propagate plants, multiply plants. So, and also it is very much effective for those propagules which are very difficult or which basically slow grower. For example, narcissus, all bulbous crop, even the gladiolus is very slow growing. So for that, micropropagation can be an alternative. And also you can introduce disease-free new cultivars. And last, the breeding cycle can be shortened, definitely, because you are doing the culture throughout the year. So breeding cycle is obviously shortened as compared to the field condition. But there are certain limitations. As I mentioned, just like plant tissue culture, it requires sophisticated facility, high production cost until and unless you are doing large production. So in that case, if you do a large production, then the rate of the or the cost of the each plant would be reduced. And it requires a skilled technician. But somaclonal variation is basically a problem. Somaclonal variation, as I mentioned, it's basically a variation that is suddenly it arises in, or you can say it's basically a mutation, spontaneous mutation that leads to the change in the genetic composition. So that is called the somaclon variation. The somaclon variation basically a limitation, because in case of micropropagation, definition is it should be true to type. So if somaclon variation arises, that means those plants are not true to type with the mother plant. So every time you have to be very cautious, you have to check their genetic fidelity. And hyperhydricity, or that is called vitrification, that is also a problem. Next slide I will explain. Also poor acclimatization. Sometimes what happens, you propagate large number of plants, but the acclimatization rate is low. So that leads to the finally you will get less number of plants. Okay. And for many valuable tree species, these are not possible. I mean, so far, for example, mango, it's very difficult to micropropagate mango plants through normal conventional this plant tissue culture technique, because these are very slow growers. And so these are some of the limitations. So next slide I will show, this is called hyperhydricity or vitrification, because here what happened, the tissue raised plants, these are basically hydrated. You can find this is the cells are in a hydrated state. So this is basically a physiological malfunctioning. So that results in excessive hydration, and that leads to a very low lignification, so that, and with impaired stomatal function. So basically it reduces the mechanical strength of the tissue raised plant. So this is one problem with the plant tissue culture, but there is no hard and fast I mean the reason why it happens, but some points are there. The gelling agent may affect that, maybe some hormone or like that, or even the water potential is also responsible for such hyperhydricity. So as I mentioned that it should be a virus free. So virus free, to raise virus free, usually meristem tip culture is used as a explant. So meristem, you can find here the structure. This is the apical dome, and this is the one or two leaf primordia. So this part is known as apical tip or the meristem tip. So if you excise this tip and if you use it as a explant, so there is a chance that is almost virus free. So the reason, because the reason why the virus they fail to invade this meristematic zone is, one of the reason is very high auxin concentration present at the meristematic cell, because auxin is there and at a very high concentration. And so competition for the nutrient and enzyme is going on with the plant and the virus. So virus is unable to replicate. And also since meristematic at the meristematic dome, there is a very active metabolic process is going on. That's why this is not suitable for virus multiplication. So these are some of the examples, some of the reasons why plant virus, they basically fail to invade this apical tip meristem. So that's why apical tip meristem is usually used as a explant for raising the virus-free plant. So, but how we will detect virus? Because sometimes what happens, we are doing the plant tissue culture, but since your material may be virus infected. To detect virus, plant virus, these are the different techniques. One is called physical property-based technique. You can use electron microscopy, or you can use different PCR technique, that is basically a nucleic acid based technique, or you can use viral protein property based technique, that is, or serology based detection technique. One of them important one is ELISA. ELISA is nothing but it's an enzyme linked immunosorbent assay. That is a biochemical assay that is used for detection of plant viruses in the tissue raised plant. So advantage is very sensitive and also very cost effective. You see that a large number of plant sample can be tested within a very short time. But detection is, since this is basically antigen antibody reaction is there, it is based on the presence of the antigen. So unknown, the virus of unknown origin is very difficult, because you have to design antibody, then you have to apply the plant extract having that virus. So in case of virus of unknown origin, it is very difficult to know the nature of the that coat protein. So it is difficult for ELISA to detect unknown viruses. And also that viroid, this is basically there is no coat protein is there, so you can't detect viroids using the ELISA technique. So this is a ELISA plate you can find. This is called microtiter plate, and this yellow color, it means positive result. That means here there is 96 wells are there. That means you can screen 96. You can screen 96 random plant samples. And if you so you can find these yellow color means this is a positive result. That means in those wells the plant extract or the plant is basically, or the plant material, that is either you collect it from the tissue raised plant or direct from the cultured vessel, so that is a virus infected, so that is called the positive result. And all the colorless one is basically the negative result. That means there the plant samples are not infected with virus. So this is basically three different modes are there, or direct ELISA, indirect ELISA, and sandwich ELISA. I'm not going into details. So these are some techniques by using you can detect the virus, presence of virus in the plant material. So because you have to ensure, once you are taking an order to deliver maybe 2 lakhs plant, that should be virus free. So you have to randomly check your tissue raised plant for using this ELISA kit, or so that you have to ensure that your plant material should be virus free. Another aspect is you can raise haploid plants. That's very important, because you can, if you select X plant as anther or the pollen, then it's there is high possibility that you can raise haploid plants, because these are in haploid structure. So haploid plant production is one of the advantage of using plant tissue culture. And once you know that haploid plants is basically a weaker, usually these are smaller and they have very weaker plant, they are sterile. So it is always desirable to multiply or to double the chromosome number by using the colchicine. So once you double the chromosome number of a haploid cell, that is that is originated through this raised through the plant tissue culture technique, so you will get double haploid. So double haploid is one of the, you can say, the it's now it's become stable. Here on the left side, the E zone, you can find this is basically that chromosome number is n equal to 21, but after multiplying the doubling, you are doing the chromosome number doubling using colchicine, the chromosome number has doubled. Now it is 42. So this is called the double haploid. So once it is double haploid, that means the plant is stable and there becomes fertile, because once it is in a haploid state, so during the meiosis, so, or you can say if you see the at anaphase stage, so random distribution of the chromosome will happen. So that leads to non-viable formation, non-viable gamete. But once you double the chromosome number, so there is normal segregation of chromosome takes place at anaphase 1. So it will form the viable gamete. So finally you will get a viable plant, or you can say as compared to these you can say that the haploid plants, you can raise very stable diploid plants by using this by applying the colchicine. And so you can find the double haploid. The advantage of raising this double haploid is within a very short time, because within 2 years you can raise a homozygous line. Okay, because homozygosity can be achieved within 2 years, because first year you can dedicate for the raising of the haploid plant. In the next year you can just double the chromosome number using the colchicine, so you will get the double haploid. So once you get the double haploid, that means you will achieve the homozygous line. Whereas if you see conventional breeding technique, it takes 5 to 6 years to develop these homozygous line. So as I mentioned, there is a chance of somaclone variation. So somaclone variation or the change at the genetic level can be checked using different markers. So DNA markers is very powerful and sensitive technique. So these are the classification of DNA markers. Some are probe based, for example RFLP, restriction fragment length polymorphism. Some are amplification based, maybe PCR based, it may be RAPD, SSR, ISSR, like that. And some are combinations of this PCR based and a probe based, that is AFLP, amplified fragment length polymorphism. So these are the dominant, co-dominant. I'm not going into details. So this is a PCR. So what happened if you raise maybe the 10,000 plant in one batch, you have to randomly collect the plant sample, you have to randomly isolate the DNA, and then you have to do the this RAPD. Maybe you can run the RAPD. So sometime what happened that out of these 100 sample, maybe one or two sample, there is either absence of a band or maybe presence of an extra band. So that means there is a change at the genetic level. So here also you can find the sample 2, here the B band is absent. That means here some genetic change has occurred. What change has occurred? Because here you can find that the change basically leads to the primer binding site has been mutated. So that is one reason. So by using different molecular techniques, particularly the DNA markers, you can detect such type of polymorphism or such type of variation. So once you find any variation, you have to discard the whole lot, because micropropagation, yeah, the raised plant should be genetically identical. So using these molecular techniques, you can identify whether there is any somoclonal variation is there or not, or any genetical change has occurred or not. So this is RFLP, again another technique. So these I'm not going into details. So just last part. So these are the basic steps of plant tissue culture. So it starts with a washing of. It starts with a selection of explant, or you can say the collection of plant material. So plant material either you have to collect from nature, or you can collect it from maybe from the field, or it may be from the market, or like that. So after collecting the explant, or you can say it's better to say the plant material, then you have to select the explant, which part you are going to inoculate. It may be a leaf segment, it may be a protoplast, it may be a node with axillary bud, or it may be the internode segment, or it may be the root, or it may be the any part, it may be the anther, or like that. So it depends on your objective. If your objective is to raise haploid plants, then you have to collect either anther or pollen or ovule as a explant. But if your objective is to multiply more number of plants, it is very obvious you can use nodal explant with axillary bud. So that will grow and multiply. So first step is collection of plant material. Second one is the selection of explant. The third one is the washing of this explant. So usually non-ionic detergent is used. It is just like a hand washing. Once you enter your room from outside, we used to wash our hand with a liquid detergent. So here the detergent should be non-ionic. So 2-in-20 is very common. That detergent is basically used to wash the explant. Then the next step is the surface sterilization. Surface sterilization is done using some chemicals. Some are basically not good for the nature, very much, corrosive and also toxic for the nature. That is HgCl2. Previously, it is being used in every lab. But nowadays, sodium hypochlorite is used, which is basically less toxic. So after doing the surface sterilization, you have to just put a ethanol wash. Then you have to inoculate. Inoculation means that once the surface sterilized explant is inoculated in a media. So that media, I will tell the composition of the media in the next few slides. So the first step is collection of plant material. Second one is selection of explant. Third one would be washing of the explant. Next would be the surface sterilization. Next would be the ethanol wash. Next would be the inoculation. So once you inoculate, then you have to keep if it is in a petri dish. Here you can find this, the culture is being made on petri dish. So if you culture in a petri dish, or in the culture tubes, or in flask, then you have to keep it in the rack, where you have to maintain that artificial condition. And so once after some time, it may be after 7 days or maybe 10 days, it depends on the explant. Or if you wish to initiate callus, it takes a long time. Whereas if you see whether these axillary bud grows or not, after maybe 4 or 5 days, you can find if there is no infection, you will find that axillary bud it grows. So the next step would be regeneration of shoot followed by root, then hardening, and then you can shift it to your net house and like that. So this is basically some steps of normal plant tissue culture. So what are the requirements for doing a plant tissue culture? There should be, for establishment of a plant tissue culture lab, there should be a separate media preparation room where the media should be prepared. There should be a separate washing room so that you can wash the culture vessels or utensils and everything. There should be an aseptic transfer chamber area there should be a environmentally controlled culture room. That means the culture room where everything is controlled artificially. It may be the temperature. The temperature is being maintained by using air conditioner. Usually two air conditioners are installed in alternative in a series, so that using a timer, one may operate for the four hours, the next one may operate for the 4 hours, like that. So it maintains the temperature of that culture room at a 25 degrees centigrade. It should be plus minus one, like that. And here you can find on the upside of the rack there are fluorescent tubes are there, so that basically maintains the photo period. So there are certain timers are there which are being installed, which basically regulate the circuit. So it is usually 16-hour light, 8 hours dark. So that basically mimic the photo period. The humidity should be maintained 50 to 60%. So like that. So apart from that, there is you require an analytical room. That means there you have to weigh your chemicals, your the inorganic salts and all these things. There should be acclimatization room. The acclimatization room is very similar. Only difference is the plant is now outside the out of this culture vessel. So rest part is it is same environmentally controlled chamber where the temperature is being maintained at the same centigrade, that means 25 degrees centigrade, with the same photo period or like that, everything. And some miscellaneous items are required. So these are basic lab instruments are required. One is very important, pH meter, because the media pH should be 5.6 to 5.8 because it is required for optimum absorption of the nutrient from the media. And also it basically helps the media. Otherwise, if there is an error in the pH, no, so there is some gelling problem is can be happen. So you require very sophisticated weighing machine so that you can weigh your inorganic salts. You need an electronic hot air oven. It is also required for sterilization. You need microscope, mostly the stereo microscope, to observe whether there is any development of somatic embryo or not, should what is there. So you need centrifuge, you need filter sterilization, and most important, you need this laminar airflow or laminar hood, because in the first slide I mentioned that you have to culture each and every part, whatever they explant, on nutrient media but under aseptic conditions. So laminar airflow is that cabinet where it provides that ultra clean air. So these are some common, instrument, or you can say the utensil that are being that is basically required for culture initiation. It may be simple scissors, scalpel, different size of forceps. So here you can find the second photograph. You can find this student. This basically the one group has come from nearby school. So this girl, she is doing a culture. So here you can find, because it happens just after lockdown. So everyone with this mask. So this cabinet is known as this laminar airflow or the laminar hood. On the third panel you can find. So this particular cabinet is also being used in case of microbiology, juvenile pathology. So this cabinet is used to provide that ultra clean air, or this aseptic condition is basically maintained under this hood. So it has a specific HEPA filter. You should remember the term HEPA. HEPA means high efficiency particulate air. Basically it removes the particles larger than 0.3 micrometer. That means it has a very small pore size. So once the air comes through this pore, this HEPA, so it is expected it gives a ultra clean air which is free of fungal or bacterial contaminants. Okay, so it is free of fungal spores and bacteria and like that. So the efficiency of this particular laminar hood, it depends on the quality of the HEPA. So based on the HEPA, so the price of this cabinet, it depends. So these are the different aseptic techniques. So first one is a sterilization I mentioned. So sterilization means that is the complete destruction of the pathogen or the living materials. Okay, but disinfectant, it basically, these are the chemical agent used to kill pathogens, but without sterilizing the matter to which chemical is applied. That means if you do disinfectant, it will only kill the pathogens. Not if you apply disinfectant on the table, so that will not lead to the sterilization. But if there is certain pathogen, that will be killed. And sanitization, it is basically it basically substantially reduce the overall load of the microorganism in that. So that because during the lockdown we used to say that sanitization is going on like that. So sanitation is nothing but it will not completely destruct or completely, or you can say the destroy these living materials or the pathogens, but it reduces the load of the microorganism load. And these are some of the chemicals commonly used for sterilization. As I mentioned, nowadays sodium hypochlorite is being used. Previously, this mercury chloride was used, but it's very toxic for the nature. That's why in abroad it is basically banned. So we are now using the sodium hypochlorite or any other that is mentioned here. So these chemicals are used for sterilization of the explant. But here you have to also sterilize your glasswares, plastic wares, and all the media. No, so dry heat, which is can be achieved by keeping your glasswares, clean glasswares, into the hot air oven, and you have to keep it for 160 to 180 degrees centigrade for 3 hours. So that will give you a sterilization. Basically, this is for the glasswares. Okay, so not for the plastic wares. There is another mode of wet heating that is basically known as autoclaving. You know that autoclave. So this is basically, here I have shown, this is an instrument that basically called autoclave. So this is nothing but a big version of a normal pressure cooker. Here what happened when the volume is fixed, when you heat that coil, so there is a water vapor generated, so it basically increases the pressure. So usually 15 psi pressure is being maintained, and that is correspond to almost 120 to 121 degrees centigrade. And you have to keep the material, particularly the this culture media, and along with the other, maybe some water, maybe some other utensils, you can keep it for autoclaving. So the time is it depends on 15 to 45 minutes, and also volume of that media is also very much important. Based on that, you can manipulate your autoclave time. If it is a very large vessel or very large quantity of media is there, so in that case, usually 30 minutes or 35 minutes or 40 minutes is usually do this autoclaving. And sometimes some chemicals are basically, particularly hormones, these are thermolabile, not thermostable. So if you autoclave it, so it will lose its property. So that's why this type of filter sterilization is done, because here there is a similar like HEPA, there is a filter is there. So its pore size is very small. So when you pass the you can say the non-sterile hormone, it maybe the GA3 gibberellic acid solution. So it will go through this filter. So since the pore size is very small, so the filtrate is basically free of any pathogen. So for thermolabile, you should remember the filter sterilization is done. And so these are the different types of media which are available. So most important media is Murasik Skoog. It is basically formulated in 1962. Apart from Murasik Skoog, you can find there are other media there. There is a Gambog, it may be white, it may be Mitra, and like that. Whatever the name of the media, but its general composition are some inorganic salts, some vitamins, some growth regulators, some carbon source, organic supplement, and gelling agent is optional, as I mentioned. So it could be, it may be solid or it may be liquid. So this is just I'm showing the composition of Murasik Skoog media. It's 1962 proposed by Murasik and Skoog. Some are macro salts. That means these salts, which are maybe ammonium nitrate or maybe potassium nitrate and so on, those are required in a very large quantity. That means you can find this is 1650 mg per liter. That means for preparing 1 liter media of Murasik Skoog, you have to weigh 1.6 gram of ammonium nitrate or like that. But there are certain microsalts are there. For example, boric acid, it requires only 6.2 milligram per liter. So mostly what happened for microsalt, we used to prepare stock solution, stock strength. So that, because sometimes what happened here, you can say potassium iodide, it requires only 0.83 mg per liter. So if you are preparing 250 ml of media, it's very difficult to weigh this very small amount of, because this should be divided by 4. So it's very difficult to weigh potassium iodide using a normal balance, normal or high sophisticated balance. So for that, it is advisable to prepare stock. So there should be iron source, there should be vitamins should be there, And carbohydrate is usually sucrose is used. 3% sucrose we use. Some amino acid supplements should be there. So these are the different properties, and if what are the advantages and disadvantages of these using these elements, or the what the outcome of deficiency of such elements is there. For example, if you see that in case of magnesium, here I have shown this one. So you know the magnesium is in the center of the chlorophyll. So if there is a deficiency of this magnesium in the your nutrient medium, there might be chance that your leaf becomes yellow. So each and every element has some role, particularly these minor elements. So these are some organic compounds are there, some growth regulators. So I'm not going into details. So this is an activated charcoal we used to apply sometime in the media, because for those plants which have high secondary metabolites, so it's the media becomes, or the plant material becomes brownish in color. So we used to apply activated charcoal, a very small amount, 0.5 gram per liter. So it basically absorbs these inhibitory compounds or these secondary metabolites. So that's why it is advisable to use activated charcoal in the media where you are using an explant, which usually it's known for its very high concentration of secondary metabolites, where when there is a high concentration of secondary metabolite, it is always advisable to add this activated charcoal. So this much I should stop here, because it's almost more than 1 hour. So student, if you have any query, you are free to ask. So I would be happy to give you answer.
Thank you so much, sir.
Yes,
yeah, it's very fascinating, and in a very lucid way you have delivered your lecture, and exactly what the student need for the UG studies. So student, do you have any questions, and faculty member also, you can always ask or reach out to the sir for any clarifications, because you have delivered it so lucidly so that you can understand the basic thing about this tissue culture system, the kind of the media or the type of what should a tissue culture lab should be, and what kind of instrument it should have. So any questions do you have, you can easily ask, sir.
Yes, student. If you have any query, even you can ask in Bengali, so don't worry.
Yeah. In any language you can ask.
Yes. I think it is in their 6th semester syllabus, no?
Yeah, mostly in the 6th semester syllabus they have, sir. Mostly in the 6th semester, because most of them are from the 4th and the 2nd semester. But yeah, it's in the 6th semester mainly.
Yes,
So,
actually the whole syllabus in 6th semester, it is covered in Zahed sir's lecture. So I asked students of 6th semester, particularly Piu and Rakhi, if you have any questions, you can ask Zahed sir. And this seminar actually is for you. Particularly in the plant tissue culture part, it is totally your syllabus oriented. Zahed sir, what about told you, totally it is syllabus oriented, KNU syllabus. Zahed sir has delivered the lecture in a very lucid way, and the total full lecture was the syllabus oriented. Okay, so I think that you have got interest, and if you have any questions, you ask to sir. If you ask if you have the questions, you can ask in Bengali also. Although Bengali is not opted in your curricula, but you can ask. One of the participant students, although he is the first year student, first semester student, he put a message that if the lecture was in Bengali, it would be feasible for him. But although he has not acquainted with such type of the tissue culture technique right now, as it is not included in the syllabus of the first semester. Anyway, but the 6th semester students and the 4th semester student also, if you have any questions, you can ask.
Hello, sir.
You Rakhi, question nahi?
Sir, I have a question, sir.
Okay, Otanu. Okay. Otanu, please.
Sir, can we conserve endangered plant species through tissue culture method?
করা হয়. So that is because, as I mentioned, that is a vast area of. So it's very. It's not possible to accommodate all these things under one our lecture. So obviously, plant tissue culture can be used to preserve endangered plant species, and that is being used nowadays. Because sometimes, but you have to establish or you have to standardize the protocol. Until and unless you standardize the protocol for that endangered plant species, you can't do tissue culture, because in any protocol, if it is applicable for maybe the rice plant, it is not mandatory that it will be very much applicable for the wheat or like that. So if you standardize the protocol, you can maintain and you can propagate those plants. Even for the taxol, if you see this taxol is very much now it is very high demand, no? So it is also prepared through this secondary metabolite concept is there, no? So if you do this micropropagation and if you extract taxol in large quantity from that tissue raised part, so this is possible. So, but it all depends on the standardization of the protocol.
Okay, sir. Thank you.
অতনু আমি একটু তোমাকে অ্যাড করে দিচ্ছি জাহেদ স্যারের সাথে। জাহেদ স্যার তো তোমাকে ডিটেলস ওই ব্যাপারটা বললো, আর আমি একটু সাধারণভাবে যেটা অ্যাড করছি সেটা হচ্ছে এন্ডেঞ্জার স্পিসিস গুলো সাধারণত যেটা হয় ওদের রিপ্রোডাকশনটা ঠিকঠাক হয় না, রিপ্রোডাকশনের একটু ব্যাহত হয়। এবার রিপ্রোডাকশনটা মানে পলিনেশন এবং রিপ্রোডাকশনটা ঠিকঠাক হয় না। কেন হয় না? এনভায়রনমেন্টাল ডিগ্রেডেশন, হ্যাবিটাট ডিগ্রেডেশন, বিভিন্ন কারণে, এখন এই গ্লোবাল ওয়ার্মিং, ক্লাইমেট চেঞ্জ, এই সমস্ত কিছু কারণে দেখা যাচ্ছে অনেক কিছু স্পিসিসের তাদের রিপ্রোডাকশন ব্যাহত হয় এবং রিপ্রোডাকশন ফেলিওর হয়। পলিনেশন, সাকসেসফুল পলিনেশন রিপ্রোডাকশন যদি ফেলিওর হয়ে থাকে, সেটা বিভিন্ন লেভেলে হতে পারে, স্টিগমেটিক লেভেলে হতে পারে, স্টাইলার লেভেলে হতে পারে, পোস্ট পলিনেশন স্টেজে গিয়ে তোমার ওভারি স্টেজে গিয়ে কোনো ফেলিওর হতে পারে, বিভিন্ন কারণে অ্যাবর্শন হয়ে যেতে পারে, পোলেন অ্যাবর্শন হতে পারে, ওভিউরের অ্যাবর্শন হতে পারে, বিভিন্ন কারণে তার রিপ্রোডাকশন ফেলিওর হবে। তো রিপ্রোডাকশন ফেলিওর হলে পরে কি হবে? তার ফ্রুট সিড হবে না। তো ফ্রুট সিড না হলে পরে সেই গাছটা মরে যাবে, আলটিমেটলি তার প্রজেনি থাকবে না। তো তখন সেই প্রজেনিটাকে বাঁচাবার জন্য আমাদেরকে শরণাপন্ন হতে হয় জাহেদ স্যারদের, মানে টিস্যু কালচার। ওনারা করেন কি, ওই ভেজিটেটিভ সেলগুলোকে নিয়ে, তার লিফ এক্সপ্ল্যান্ট বা কোনো রুট এক্সপ্ল্যান্ট, ওই ভেজিটেটিভ সেলগুলোকে নিয়ে বায়োটেকনোলজি মেথড করে সোমাক্লোনাল ভেরিয়েশন টেরিয়েশন এই সমস্ত এনে ক্যালাস কালচার করে তোমার সোমাটিক এমব্রায়ো তৈরি করে ওনারা তখন ওই গাছটাকে নতুন রূপ দান করেন, নতুন ভাবে গাছটাকে দেন। বুঝতে পেরেছো? তো এইটার জন্যই মেনলি কিন্তু আমরা টিস্যু কালচারটাকে খুব ইজি একটা টেকনিক, প্ল্যান্ট বায়োটেকনোলজির একটা টেকনিক, যেটাকে করে কিন্তু আমরা ওইগুলোকে ওই এন্ডেঞ্জার স্পিসিস গুলোকে কনজার্ভ করতে পারি। বুঝতে পেরেছো?
ওকে স্যার, ওকে স্যার, থ্যাংক ইউ।
আর কিছু বলো?
আচ্ছা, স্টুডেন্টদের উদ্দেশ্যে বলছি যারা এখানে আছো, তাদের একটা আমি গুগল লিংক শেয়ার করেছি, ওইটা তোমাদের অ্যাটেন্ডেন্স, ওটা অ্যাটেন্ডেন্সটা ফিডব্যাক অ্যাকচুয়ালি রাদার ফিডব্যাকটা ইফ ইউ প্লিজ পুট অন দ্যাট ফিডব্যাক অ্যান্ড ইউ উইল গেট ইউর সার্টিফিকেট অটোমেটিকলি। হ্যাঁ, তোমরা নিজের সার্টিফিকেট অটোমেটিকলি পেয়ে যাবে যদি ফিডব্যাকটা ফিল আপ করো। সো প্লিজ fill the feedback form as soon as possible and the link is in the chat box. So anything else, student, if you want to ask, so I guess not, then I will request Mr. Anish Bhattacharya, one of your students, sir, to give you a formal vote of thanks. Anish, are you there?
Am I audible?
Yeah,
Yes, yes.
you're much audible, please, Anish.
Hello sir, how are you?
Fine. How are you, Anish?
অনিশ, তোমার ভোট অফ থ্যাঙ্কস হয়ে গেলে আমাদের প্রশান্তর সাথে একটু জাহেদকে কথা বলিয়ে দিও, আমাদের ফ্যাকাল্টি প্রশান্ত তো বায়োটেকনোলজি।
So good afternoon, our esteemed speaker Dr. Zahed Hossain sir, respected principal sir, respected faculty members, and my dear fellow participants who have participated in the one-day invited lecture named Modern Techniques of Molecular Biology and Plant Biotechnology for UG students. Thank you, Dr. Ray, for giving me the opportunity to give the vote of thanks. I feel really lucky to give vote of thanks to our respected speaker, sir, whom I really admire and who is one of my favorite teachers and guides who have advised me in every aspects of life. I would like to extend my sincere thanks to sir, who shared his expertise and experience with us. His presentation were enlightening and inspiring, and we are very grateful for his valuable contributions. I would like to express my heartfelt gratitude to our Honorable Principal Sir, Dr. Devnath Pali, for gracing today's lecture. Thank you, sir, for your very thought-provoking and interesting address. Your insights have truly enriched our understanding of the fascinating field of plant tissue culture. I would like to thank our respected Deputy Coordinator, Madam Dr. Nivedita Acharji. I'd like to thank our respected Head, Department of Botany, Dr. Ashok Bhattacharya, sir, for his excellent introduction of our Honorable Speaker. Secretary of Teacher Counseling, Dr. Shubhajit Oza, for his motivating words. A very big shout out to our organizing department, Dr. Sondipon Ray, Dr. Shubhajit Oza, Dr. Prashanto Sayan, Mr. Dibyendu Midda, who worked so hard behind the scenes to ensure that every aspect of the seminar ran smoothly. Last but not the least, a heartfelt thank you to all our fellow participants. Thank you for your active participations and engaging discussion. Your presence here today signifies your interest in the biotechnology, and we hope we found this seminar both informative and enjoyable. Thank you so much.
Thank you, Anish.
থ্যাংক ইউ অনিশ। থ্যাংক ইউ। এই প্রশান্ত,
প্রশান্ত দা, তোমার তুমি
হ্যালো স্যার। না ঠিক আছে, স্যারের সঙ্গে স্যারের লেকচার আজকে ফার্স্ট শুনলাম, ভালো লাগলো, প্রচন্ড এক্সটেন্সিভলি স্যার বললেন এবং খুবই ভালো মতো ভাবে খুবই লুসিড ওয়েতে বললেন। মানে স্টুডেন্টরা রিয়েলি খুব বেনিফিটেড হয়েছে স্যার। থ্যাংক ইউ সো মাচ।
আসলে এটা অফলাইন হলে একটু বেটার হতো, অ্যাকচুয়ালি অনলাইনে না ইন্টারাকশনটা ওয়ান ওয়ে হয়ে যায় তো।
এই জাহেদ একবার অফলাইনে একবার নিয়ে আসবো, দাঁড়াও না, সুযোগ সুবিধা হলেই একবার তোমাকে এটা একটু
ঠিক আছে
একবার চলে আসবে, সবার সাথে একটু ঘুরে বেরিয়ে যাবে, দেখা যাবে।
মানে
তারপরে হচ্ছে থার্ড আর্টিকেলস এ আদার মলিকুলার টেকনিক্স এবং হেন্স লাস্ট আর্টিকেল হচ্ছে ট্রান্সজেনিক প্লান্টস। তো এন্টায়ার আর্টিকেল 1 টা ওয়াজ এক্সটেন্সিভলি কভার্ড স্যার। মানে এটা আমার খুবই ভালো লাগছে। মানে আসলে আমিও ধরুন 1.5 মাসের মতো এফআইপি তে ছিলাম, তো সেই জায়গা থেকে একটা গ্যাপও হয়েছিল আর কি। আমাদের আরেকজন স্যার উনিও নেন ক্লাসগুলো, তো স্টিল আপনি যেটা বললেন মানে ওদের মানে প্রচন্ডই বেনিফিটেড হবে আই সাপোজ। কি পিউ রাখি মানে আই সাপোজ মানে ইউ হ্যাভ বিন বেনিফিটেড টু আ গ্রেট এক্সটেন্ট বাই দিস লেকচার।
মানে এই পেপারটা জাহেদ আমাদের এই প্রশান্ত পড়ায়, আর এবছর আবার আরেকজন শেয়ার করেছে শুভজিৎ, তার টিস্যু কালচারটা বোধহয় সম্ভবত শুভজিতের শেয়ার করা ছিল আমার যেটা মনে পড়ছে। তো শুভজিৎ এবং প্রশান্ত দুজনে মিলে এবছর পড়াচ্ছে। অ্যাকচুয়ালি এটা প্রশান্ত পুরো পেপারটা পড়াতো তো, এবং এটা তুমি যেটা আজকে বললে এই যে 1 ঘন্টা একটা লেকচারে না বিরাট উপকার হবে আমাদের 6 সেমিস্টারের স্টুডেন্ট গুলো, কারণ পুরোটা কিন্তু একটা ইউনিট ওদের কভার হয়ে গেল এই সিলেবাসটার মধ্যে, এবং তুমি সেইগুলোই বলেছো সমস্ত। এটা তোমাকে অনেক ধন্যবাদ ব্যক্তিগতভাবে। আর এরপরে তোমার সাথে সন্দীপন সব যোগাযোগ করে নেবে সমস্ত কিছু অন্যান্য টেকনিক্যাল ব্যাপারগুলো যেগুলো আছে সেই সব ব্যাপারে।
ঠিক আছে,
একটুখানি পুট আপ করতে চাইবো আমি স্যারকে, আরো স্পেশালি থ্যাংক করতে চাইবো এই কারণে যে আমাদের ধরুন স্যার বেশ কিছু প্র্যাকটিক্যালস মানে এখন কলেজ যেহেতু টেক ওভার হয়েছে, মানে এই সময় দাঁড়িয়ে একটুখানি সমস্যা হয়েছে। তো সেক্ষেত্রে আপনি যে একদম রিয়েলিস্টিক ইমেজেস এবং প্রত্যেক কটা ছোট থেকে বড় ইউটেন্সিলস থেকে শুরু করে কিভাবে কি হচ্ছে ল্যামিনার, মানে আমাদের ডিপার্টমেন্টে রেনোভেশনও চলছে, টিস্যু কালচার রুম একটা ডেভেলপ করা হচ্ছে আমাদের এখানে। সো তা সত্ত্বেও আপনি যে দেখিয়েছেন এত ডিটেলড ওয়েতে, এতে স্টুডেন্টরা আই বিলিভ মানে দে হ্যাভ লার্নড আ লট স্যার। থ্যাংক ইউ সো মাচ স্যার।
Thank you Ashok da, thank you Anish,
thank you bhai, thank you
so much sir, thank you so much,
thank you sir. So we are ending this session. We will hope to meet you again soon sir,
in person. Thank you Sandipon. Okay, bye.
Bye.